very pleased to have Dr. Jayaraman. He's PhD, coming to us from University of Illinois College of Medicine. And the title of his talk is Epigenetic Regulation of Type 1 Diabetes. A little bit about him. After receiving a PhD from Madhuri Kamarja University in India, he completed his postdoctoral training in immunology at St. Louis University, and he held faculty positions at Harvard University, Washington University, St. Louis, and the University of Miami, now at the University of Illinois in Chicago. And he studied T cells involved in asthma, type 1 diabetes, multiple sclerosis, and herpes virus-mediated stromal keratitis. Using animal models, his team investigated the impact of epigenetic modulation, specifically histone hyperacetylation, on T cells involved in autoimmune diabetes. The whole genome transcriptome analysis revealed the correlation between the overexpression of a selected set of genes and the manifestation of autoimmune diabetes. Treatment with a potential histone deacetylase inhibitor repressed the exaggerated genes and afforded protection against type 1 diabetes. These data suggest that epigenetic modulation can represent a novel treatment modality for type 1 diabetes. His newest paper is, I'm dropping in the chat. And welcome, Dr. Jayaraman. We're very happy to have you with us. And if you'd like to proceed and show your slides, that'd be great.
Thank you, Dr. Wesley, for this invitation. I'd like to, okay. Can you see
Yes,
that?
Looks great.
Okay, so I'm from the University of Illinois at Chicago. The work that I'm going to present is from University of Illinois at Chicago and later in Peoria. Okay, this is just to give you an idea of our contribution to this area of epigenetics in modulating autoimmune diseases like type 1 diabetes and also multiple sclerosis. And we also had worked on tumor models to see the epigenetic regulation, how it can be used to treat and screen diseases. Just to give you an idea of what is epigenetics, it's defined as the study of heritable changes in genome function that occur without a change in DNA sequence. So this is apart from genetic variants that have been described in many autoimmune diseases, including type 1 diabetes. The idea of epigenetics goes back to Aristotle period, and then actually Conrad Paddington was the one who popularized this concept. Now it's generally accepted that gene expression can be modulated by epigenetic mechanisms. There are several epigenetic mechanisms described. I'm going to focus on one type of epigenetic modulation, that is the histone modification of by a lysine residue at the tail of the nucleosome. And that can be done by a number of methods. To give you a brief introduction about the type 1 diabetes, as you know, it's one of the 80 or so autoimmune diseases known to human kind. Specifically, type 1 diabetes is a very common childhood illness. It co-occurs with a variety of autoimmune diseases, including thyroid diseases and multiple sclerosis, to name a few. And the major contributors of this type 1 diabetes is the susceptibility genes encoded by MHC class 1 and 2 antigens. And although there have been some speculations about what certain viruses can contribute to the initiation of the disease, but it's not well proven. And there has been speculation about the role of gut microbiota, which we actually have recently analyzed and published in 2024. So the bottom line is that all these contributors can affect type 1 diabetes via CD4 positive T cells and the macrophages. These two cells are critical for the destruction of insulin producing beta cells, as shown in the right-hand side top corner. So in the absence of beta cells, the patient will require exogenous insulin for survival. And as a result of epigenetic modulation, like the one I'm going to present data on, is the histone modification. The disease will be retarded. I wouldn't use the term reversed, and it accompanies a lot of gene regulation. We chose the small molecule inhibitor called trichostatin A, which was described by a group of Japanese investigators in the 1980s, which was derived from Streptomyces species, and it was originally described as an anti-fungal agent. And for the past 20 some years, this drug has been studied very widely, and it's now known that it inhibits. Originally, it was described that the TSA can inhibit a variety of hashtag genes, which are histone deacetylases. And we have shown recently that this drug can also inhibit the class 4 hashtag, which is hashtag 11. So except the class 3 sequence, and all the other hashtags are susceptible to the action of this drug. And it has been shown that the TS administration can protect, at least in the mouse model, against type 1 diabetes, which I'm going to talk about. And EAE is a model of multiple sclerosis, which we have studied recently. And others have described the protective action of TSA on a variety of diseases like lupus, asthma, arthritis, ALS, glioblastoma, and many tumor models. We started this work back in 2011, and we injected the NOD mouse, which is an excellent model to study type 1 diabetes because it reflects most of the events that occur in type 1 diabetes, not exactly the same. Human and mouse are departed like 60 million years ago. So there are some differences. Nonetheless, this mouse, again, was derived in Japan to isolate the genes for cataract. And the beauty of this mouse is that the female mice, 80 to 90% of them are susceptible to develop the type 1 diabetes spontaneously without any intervention. So we used this model, and we injected these mice weekly for this specific period of time, between 5 and 10 weeks of the age, 10 to 18 and 18 to 24, which seems to be the most effective period in which one can intercept the progress of type 1 diabetes pathology. We studied the effect of the drug systematically to understand how this drug can interfere with the pathologic events occurring in the NOD mouse. The first thing we tested was to see if the drug can you see the full screen of the full screen? Okay. Of the data? Okay, fine.
Yes.
Okay. Yeah, I'm having trouble. Okay, so we challenged these mice after treatment of the mice with the drug, of a drug treatment, and challenge them with glucose and see if it can maintain the normal glycemia. As you can see from the left-hand top corner of the slide, the drug-treated mice exhibited normal glycemia. And it was accompanied by the secretion of insulin in the mouse about the same time, like 10 minutes. And the drug also inhibited the influx of inflammatory cells in the pancreas. This is the section of pancreas stained with insulin antibody and analyzed by confocal microscopy. As you can see, the drug-treated mice had minimal infiltration. And also we noticed that there are a lot of new islets in these mice. One of the things that we wanted to see was to see whether the drug actually induces hyperacetylation of histones. So we did the Western blotting of spleen and pancreas extract and blotted with the anti-acetyl H3 antibody. So it shows that the drug can induce acetylation of histone as we expected. And that lasts for 2 weeks. And when we analyzed them by 6 weeks, the effect disappears. So it's only transitory. The next thing we analyzed was using confocal microscopy to see whether the drug actually preferentially inhibit the influx of cells. Like I mentioned in the beginning, type 1 diabetes is mediated by CD4 positive cells predominantly, and there are some evidence that CD8 cells also participate in this model. And insulin producing beta cells are destroyed by the influx of LH6 positive macrophages. As you can see in the top panel, the diabetic mice, you can see that the cellular infiltrate is predominantly by CD4, LH6 positive macrophages, and neutrophils do not play a role in this model. And both CD4 and CD8 positive cells are decreased in the drug-treated model, indicating that this disease, like established before, is mediated by LH6 positive macrophages and CD4 and CD8 positive T cells. So we wanted to ask the question of whether or not the drug treatment can in fact reduce the diabetogenic potential of cells. So in order to do that, we took the spleen cells from the diabetic mice and from the THC-treated and the cured NOD mice after 28 to 30 weeks of injection. And we transferred the whole spleen cell, 20 million, into NOD skid mice, which are histocompatible with the NOD mice, except that these mice do not have T cells, B cells, and they lack functional macrophages, so they won't induce type 1 diabetes as the wild type mice. So when we transfer the splenocytes into these mice, you can see that they all become diabetic in about 2 weeks, and the effect lasts longer. So we established that the epigenetic TSA can in fact affect the ability of the T cells to transfer the disease in the NOD skid mice. So there are a lot of speculations in this area, whether or not the polarized Th1, Th2, or Th17 cells could transfer the disease. If we can verify that, and if you can show that the drug actually affects one population or the other, then it will be easier to manipulate the disease in humans as well. So we polarized the T cells derived from the pre-diabetic mice under defined the culture conditions described by a number of laboratories before us. And we verified the polarization of Th1 and Th2 cells. Th17 cells. Th2 cells we did not inquire because there is no much evidence for their role in type 1 diabetes in the NOD mouse model. As you can see that the polarized Th1 cells, when stimulated with the anti-CD3 antibody to ligate the T cell receptor, releases copious amount of interferon gamma, indicating that these cells are purely Th1, and they do release very little or undetectable level of IL-17. Similarly, the Th17 polarized T cells released only IL-17 and not interferon gamma. And although there has been some discussion about whether or not the Th17 cells have to transdifferentiate into Th1 cells in the immunodeficient mice in order to cause type 1 diabetes, we do not see the evidence for it. Interestingly, I should have mentioned this before this. The transfer of unpolarized T cells from the pre-diabetic mice did not cause diabetes, indicating that the T cells have to be specifically activated in order to become diabetogenic. And the transfer of Th1 and the Th17 cells caused diabetes comparably. Interestingly, the transfer of the Th1 cells caused a very intense islet infiltration, as you can see in the bottom panel. On the other hand, the Th17 transferred cells have only peri-insulitis. So combined together, we think that the Th1 cells and the Th17 cells have the ability to transfer the disease, and probably by using different mechanisms. Okay, so the next question was to see whether TSA abolished the diabetogenicity of both Th1 and Th17 cells. And so we transferred the T cells, treated or untreated cells, into the immunodeficient skid mice. As you can see that the T cells polarized from the drug-treated mice, whether it's Th1 or Th17 cells. Both of them had very minimal diabetes causing ability, indicating that epigenetic regulation can in fact abolish the diabetogenic potential of these T cells. I just wanted to verify whether these are again T cells of the specific subgroup, and you can see that the diabetic Th1 cells in the transferred mice only produced interferon gamma upon T cell receptor-mediated activation in vitro. And similarly, Th17 cell transferred mice also produced IL-17 in the recipient. Interestingly, the T cells taken from the drug-treated mice were not able to produce the respective lymphokines, interferon gamma in Th1 cells and IL-17 in the bottom panel, in Th17 cell transferred mice that were previously treated with the drug. The next question was to analyze the overall gene expression in the spleen cells after after TSA treatment. This is an assessment of about 40 some genes we looked at. This is the steady state level of of well-known cytokines. And as you can see, that the lymphokines are the ones that are mostly affected by the TSA treatment. And in the spleen, the lymphoid organ, TSA treatment actually caused the decrease in the transcription of interferon gamma, IL-17, IL-23, CCL2, and the transcription factor, AHR, aryl hydrocarbon receptor. The accessory cell-associated genes are not that well decreased by TSA treatment, since we only saw CCL2 expression was downregulated in TSA-treated mice. Like I mentioned, the only transcription factor we found decreased in response to drug treatment or AHR, aryl hydrocarbon receptor. So we also analyzed the gene expression in pancreas. As you can see that the pancreas does not express most of the lymphokine genes that we looked at in the spleen. And nevertheless, we saw the drug treatment induced the transcriptional repression of IL-22, 23, as well as CD39. Interestingly, only the FOXP3 transcription factor was decreased by TSA treatment in the pancreas. So these are the well-known genes investigated in this area. So we wanted to see whether we are missing out on certain genes by only looking at the small panel of genes. It's about 30, 40 genes. So in order to get some idea about how widespread the effect of the drug is, we conducted a transcriptome analysis. It's a non-hypothesis driven high throughput technology. As you can see that we have compared diabetic splenocytes with the drug-treated cured splenocytes and DMSO treated, the control treated splenocytes. To make the long story short, we observed by a series of analysis, only about 17 genes were highly expressed in diabetic mice. This is the whole spleen cell, uninduced whole spleen cells. So there's a difference of 17 genes highly expressed in diabetic mice, as you can see it in the right-hand side corner. The analysis shows that there are like 17 genes are highly upregulated in type 1 diabetes mice, which were subject to regulation by TSA. And we analyze each one of the 17 genes individually by qRT-PCR, because the genome analysis is not that highly specific and sensitive. So in order to get the good idea about the regulation of these genes, we utilized qRT-PCR, as you can see in the bottom panel. There's one particular gene that is CELA3B, which is expressed in high abundance in spleen of diabetic mice, and interestingly, that was the one that is downregulated by TSA treatment. And all the other genes, inflammatory genes, mostly, they are all caspases. No, I'm sorry, they're all peptidases, carboxylases, and so forth. They are not subject to regulation by TSA treatment. We also observed that there are certain genes that are upregulated in the drug-treated mice because TSA can regulate about 1 to 20% of the genes, and it can cause suppression most of the times, and also sometimes upregulation of certain genes. So when we analyze all of the genes that were highly expressed in the genomoid analysis, we saw this interesting observation that is the upregulation of like cat gene in the spleen and as well as in the pancreas, which is a gene that's involved in precursor formation of hemangioblasts. And we also saw the upregulation of ERMAP, which is a gene involved in erythrocyte development. And some of you may know that during the late stages of type 1 diabetes in animal models, there is a splenomegaly and destruction of red blood cells. And actually, ERMAP is one that will rectify the restoration of RBC. So it's all in consistent with the protective role of type 1, of TSA treatment. The important thing that nobody has looked at so far is whether the HDAC inhibitors actually cause the inhibition of the transcription of HDAC genes. There are 11 HDAC genes, like I mentioned in the beginning, described so far, and out of which only 3 HDAC genes, that's HDAC 4, 8, and 9 were upregulated in diabetic mice in the spleen, and they were downregulated by TSA treatment. So this gives us an idea about for the future direction, what hashtag gene we should concentrate on, because there has been a lot of interest in developing specific hashtag inhibitors. TSA is the most important pan-specific hashtag inhibitor. So, by discovering specific inhibitors, we can avoid a lot of unnecessary side effects of the drug. So anyway, so this gives an idea about the future targets for drug development. And the bottom panel shows that the pancreas does not express all the 11 hashtag genes as the spleen. And interestingly, TSA treatment did not abrogate the transcription of these genes in the pancreas. So we interpret this to mean that most of the effects that we see due to TSA treatment is all mediated by the lymphoid system, and probably the endocrine and exocrine pancreas do contribute very little to the disease. Unlike there are some proposition that both endocrine and exocrine pancreas do participate in type 1 diabetes development. That has to be worked out clearly. So this is the summary of all the data that I presented. So this is the model by which I explain how the epigenetic regulation can treat an autoimmune condition to convert to a healthy condition, not necessarily reversal of any disease. I believe that no disease can be reversed by treating the symptoms. So the epigenetic regulation actually can cause several things. One is gene silencing, like I mentioned, the CELA3B, which is a very prominent cytokine involved in both mouse model as well as in humans. And many lymphokines, specific hashtag genes were all silenced by TSA treatment. Interestingly, we discovered that the same hashtag inhibitor can upregulate genes like RMAP and LICAD, which are essential for a number of functions. So finally, I want to speculate on the implication of the data generated in NOD mice, because mice is mice and humans are different. We are 65 million years apart. But nevertheless, NOD mice is an excellent model to study some of the epigenetic or even the pathological mechanisms and the drug action. And those are not necessarily translatable 100% to humans. So, and nevertheless, NOD mouse model and the type 1 diabetes in humans share certain similarities, which is in monozygotic twins, there's only 30 to 40% concordance. There is only 30 to 40% concordance of type 1 diabetes, indicating that the genes are important, but they're not sufficient to induce type 1 diabetes. There are other factors that people think that it could be viral mediated, or it could be diet. Or there are so many factors that have been implicated that they think that they contribute to the initiation and the manifestation of the disease. Like I haven't mentioned that NOD mouse T cells are defective in terms of apoptosis, which is a self-tolerance mechanism that is the basis of autoimmune diseases in most of the cases. And the drug type and the drug TSA can reverse the lack of T cell apoptosis in this model. And similarly, we published a paper a long time ago showing that type 1 diabetes patients from long-standing type 1 diabetes are defective in T cell receptor-mediated apoptosis. So that is in line with the observations in NOD mouse. In other words, it indicates that the self-reactive T lymphocytes are defective to undergo apoptosis. And that's one of the reasons why there is an accumulation of pathogenic T cells in this model. This was shown in long-standing type 1 diabetes patients. And another observation that's striking is that the exaggerated expression of CELA3B in the mouse actually is replicated in the humans long-standing type 1 diabetes, which is a subject of a US patent. And we are pursuing that to see how far these genes, in addition to the Siglec gene, which has also been implicated in multiple sclerosis. That's gene present in expressed in the CD14 positive macrophages. Those two may provide, in addition to several other genes, provide a tool for us to diagnose type 1 diabetes early. And as I mentioned, there are many diseases, especially in the mouse models, have been treated with the TSA, and in addition to a number of cancers, including glioblastoma. And so it is encouraging to apply this knowledge to try to treat the type 1 diabetes as well as to screen the type 1 diabetes in asymptomatic type 1 diabetes patients, relatives of first degree relatives of type 1 diabetes patients, since we realized that the genes are important, but they're not sufficient. And I do not see there are many clinical trials on this. Nevertheless, there is one clinical trial on TSA that's been ongoing based on the original description of TSA as an antifungal agent. They're testing the efficacy of TSA treat nail fungus, I believe. Anyway, yes, it is encouraging. So it's not, people have to apply this knowledge for a treatment of type 1 diabetes.
Jay, can you unpack that a little bit, that last trial?
Yeah.
That's ongoing right now, and
Yeah.
it's an oral medication that's being given to people.
I think it's local application. It's a nail fungus. So TSA can be dissolved in organic solvent. So I don't, once you dilute it, then it is water soluble. But I don't know how exactly they do it. I just mentioned that there is one clinical trial going on, but not
It's really
on
not pertaining
any
to type
disease.
1. Yeah, it's just a
But
separate. Okay. All
yeah,
right. Thank
yeah.
you.
But TSA, otherwise it's also called it's in various trade names. So one of them has been shown to be effective in cutaneous T-cell lymphoma. So I don't know how far the trial is going, how successful they are. But I think these trials actually set the stage for future application to treat other diseases, especially type 1 diabetes. These are the contributors of this work in University of Illinois at Chicago. As you see that most of them are, except one postdoctoral fellow and myself, they were all MD and undergraduate students worked on this project and contributed to this work.
Great. Thank you very much. I'd just like to open it up for any questions. If anyone has
Okay.
a question, feel free to unmute yourself or raise your hand.
Yeah, I had a quick question. An interesting talk. This is Rohit Kulkarni from Joslin Diabetes Center in Boston. So I was just curious about the comments you made about SELA3B, because I think SELA3B is expressed, predominantly in the exocrine pancreas, and it's a chymotrypsin-like elastase molecule. So I was just curious about your thoughts on how SELA3B factors in, and you mentioned that it is expressed in the spleen, but I thought it's expressed mostly in the exocrine pancreas.
It is a macrophage specific gene in humans as well as in mouse, as far as I know.
But in the context of its expression in the exocrine pancreas, what are your thoughts on how it could be, because it's also implicated in pancreatitis?
I don't know which gene are you talking, you're mentioning, but
SELA3B.
gene, yeah, SELA3B is expressed in the whole pancreas. I mean, very little, but in the spleen, it's expressed very abundantly. And we haven't fractionated the spleen cells into T cells and macrophages to localize it. But in the humans also, we found this in the whole PBMC. So that includes macrophages. So there's no exocrine cells involved. So this is a macrophage-specific gene, as far as I know.
Okay.
Or preferentially expressed by macrophages.
Thank you.
Okay. Thanks for that question. Yeah, I mean, if we just do a quick search, the chymotrypsin like elastase 3B gene is expressed in the human spleen, but at very low levels compared to its high abundance in the pancreas. So that's like, that I think it's quite a bit different. In terms of mRNA, it's like 48,000 NTPM, and it's in the spleen. That's in the pancreas, and the spleen is 2.2 NTPM. No protein detected in the spleen, but strong in the pancreas. So maybe it's just sort of some local macrophage or something.
Yeah, that's what I
Anyone
believe.
else? Okay. I had a quick question for you.
Okay.
Let's see. Sorry, I had to open up that again. In your mind, what's the most promising epigenetic target or pathway from your studies that you believe could be realistically be modified or modulated in early stage or at-risk T1D patients?
Well, based on our data,
Mm-hmm.
NOD mouse model, the T cells, of course, are subject to regulation by epigenetic. This one is histone acetylation mediated epigenetic regulation. So T cells as well as macrophages are regulated.
And like, how can you mitigate some of the off-target effects that can come from a global medication like, TSA?
Yeah, that's why we conducted that global gene expression analysis, the transcriptome analysis, which I didn't go into much details, but we found that a few genes, not all of them, we investigated like 164 highly expressed, top expressed genes from the genomic analysis. And the only thing we found was that the lymphokine genes, of course, and some of the cytokine genes, and these unexpected LICAD and RMAP genes were regulated. So based on that, we have screened some long-standing type 1 diabetes patients using the peripheral mononuclear cells for the expression of a panel of genes. And the thing that was prominent was the expression of CELA3B again in humans, as well as another gene, SIGLEC, which is also present in the macrophage of multiple sclerosis. So there are no other genes are not regulated, like all the lymphokine that you would imagine, the IL-2, interferon gamma, IL-4, and the transcription factors, none of which is highly upregulated in the resting stage in the patient. So maybe if you induce in vitro, activate them, then there might be the way of screening this, but to screen the diabetes propensity, I think the expression of exaggerated expression of these genes are critical, but gives an easy handle to follow. Does that
Okay.
answer the question?
Any other questions? All right. Well, that was a very interesting talk. Thank you very much, Jay. I really appreciate you coming on, and we'll see where this goes.
Yeah. Okay, we are working on it,
Great.
actually.
Well, have a great rest of
That's
your
what
day.
Thank you so much
Thank
for
you.
inviting me to
Of course.
participate.
Thanks
Thank
for
you.
joining us. Bye-bye.
Bye.